Presentation on Cell, created by Adhyan Academy. Prepared by Krish Kumar, Nandini Kumari, Sally Sinha, Bhargav Rai, Aditya, under the guidance of Mrs. Mamta Mehrotra. Introduction of Cell. In 1665, Robert Hooke, an English philosopher, observed the cell for the first time. A cell is the structural and functional unit of all living organisms. It contains various organelles. All living cells arise from pre-existing cells. Difference between plant cell and animal cell. In plant cell, cell wall is present, and in animal cell, cell wall is absent. In plant cell, it has large vacuole, and in animal cell, it has small vacuoles or absent. In plant cell, it has chloroplasts. In animal cell, it does not have chloroplasts. In plant cell are larger than animal cell. In animal cell are generally small in size. In plant cell lack centrosome and centrioles. In animal cells have centrosome and centrioles. Structure of plant cell and animal cell. Structure of cell wall. It is a rigid wall. It is found outside the plasma membrane. It is non-living and freely permeable. It is made up of cellulose. The cell wall of adjacent cells are glued by the middle lamella. Cell wall has narrow pores, and these pores are called pits. The middle lamella is a jelly-like substance made up of calcium and magnesium pectates. Functions of cell wall. It gives shape to the cell. It is freely permeable to water and substances in solution. It provides mechanical strength to support the cell. It protects plant from various climatic conditions. Structure of cell membrane. It is a living, thin, delicate, elastic, selectively permeable membrane. It is made up of protein, lipids, and carbohydrates, etc. It is flexible to allow cells. The cell membrane also carries receptors. Plasma membrane encloses the border of cells, but rather than being a static bag. Function of cell membrane. Cell membrane permits the entry or exit of some material in the cell, protects the cell by acting as a barrier, receives chemical messengers from other cells, acts as a receptor. They keep toxic substance out of the cell, acts as enzymes, control metabolic process. Structure of nucleus. The nucleus is the large, centrally located, spherical cellular component. It is bounded by two nuclear membranes, both forming a nuclear envelope. The nuclear envelope separates the nucleus from the cytoplasm. The nuclear envelope contains many pores. The nuclear pores and enclose the liquid ground substance, the nucleoplasm. Structure of nucleus. Function of nucleus. Nucleus controls all the metabolic activity of the cell. It regulates the cell cycle. Nucleus is the storehouse of genes. It is concerned with the transmission of hereditary traits from the parents to offspring. Difference between prokaryotic cell and eukaryotic cell. Let's start with prokaryotic cell. Size of this cell is generally small. Nucleus is absent. It contains single chromosome. In this, Nucleolus is absent. Cell division takes place by fission or budding. Now, eukaryotic cell. Size of the cell is generally large. Nucleus is present. It contains more than one chromosome. In this, Nucleolus is present. Cell division occurs by mitotic or meiotic cell division. Structure of prokaryotic cell. Structure of eukaryotic cell. Structure of endoplasmic reticulum. Structure of endoplasmic reticulum. Endoplasmic reticulum is connected to the cell membrane towards the outer side and nuclear membrane towards inside. The tubular network is dynamic, continually rearranging and growing. The endoplasmic reticulum is an extensive tubular framework connected to cell membrane towards outer side and nuclear membrane towards inside. The endoplasmic reticulum is made up of two subunits, rough endoplasmic reticulum and smooth endoplasmic reticulum. The endoplasmic is a network of membrane found throughout to the nucleus. Function of endoplasmic reticulum. It forms supporting skeletal framework of the cell. ER provides a pathway for the distribution of nuclear material from one cell to the other. Certain enzymes present in smooth ER synthesize fats, lipids, steroids, and cholesterol. Rough ER is concerned with the transport of proteins, which are synthesized by ribosome of their surface. Difference between a smooth endoplasmic reticulum and rough endoplasmic reticulum. Let's start with rough endoplasmic reticulum. It contains flattened sacs called cisternae. It is specialized to synthesize proteins. Ribosomes are attached to the outer surface of its membrane. Now, a smooth endoplasmic reticulum. It is mainly formed of vesicle and tubules. It is specialized to synthesize of lipids and steroids. It does not contain ribosome. Structure of Golgi apparatus. Structure of Golgi apparatus. Golgi apparatus consists of a set of membrane-bounded, fluid-filled vesicles vacuoles. It has flattened sac-like structure called cisternae. It arises from a smooth endoplasmic reticulum, which originates from rough endoplasmic reticulum. Functions of Golgi apparatus. It helps in formation of lysosomes and complex sugars from simple sugars. It helps in storage and modification of products. It packages materials synthesized in the cell and dispatches them to intracellular targets. It also involved in the synthesis of cell wall, plasma membrane and lysosomes. Structure of lysosome. Structure of lysosome. They form the waste disposal system of the cell. They are a spherical sac-like structure found in cytoplasm. Lysosome is a small vesicle surrounding a single membrane. It contains hydrolytic digestive enzymes, that is, digestive bags, and is capable of breaking down all organic material. They are synthesized by the Golgi apparatus. They are also called suicide bags of the cell because if cell gets damaged, lysosome bursts and enzymes present inside it digest their own cell. Functions of lysosomes. It acts as a suicide bag in case of cell damage or if the cell is not required anymore. In case of damage to the cell, lysosomes burst and release digestive enzymes in the cell to digest the cellular components. They are thus called suicidal bags of the cell. This process is called autophagy. Lysosomes release enzymes for digestion of food. This removed unwanted and harmful substances and organism and protect the cell. Leukocyte cells dissect these foreign particles. Structure of mitochondria. Structure of mitochondria. Mitochondria is also called powerhouse of cell. Mitochondria are tiny bodies of varying shape and size, distributed in the cytoplasm. Each mitochondrion is bounded by a double membrane envelope. Outer membrane is porous. Function of mitochondria. Mitochondria are sites of cellular respiration. It produces energy in the form of ATP, adenosine triphosphate. Mitochondria are able to make some of their own proteins, so they are regarded as semi-autonomous organelles. Plastids. Plastids are double membrane bounded by organelles found inside plants and algae, which are primarily responsible for activity related to making and storing food. There are three types of plastids: chloroplasts, chromoplasts, and leucoplasts. Chloroplasts. These are green-colored plastids. They have green-colored pigment called chlorophyll. Chloroplasts are abundant in parts exposed to light. Their function is to trap solar energy and absorb carbon dioxide for the manufacture of starch and sugar. Chromoplast. These are colored plastids like yellow, orange, and red. They are mostly present in petals of flower and fruits. The xanthophyll and carotene are coloring substance associated with them. They remain dissolved in the cell sap and give red, purple, and blue colors to different parts of plant, especially fruits and vegetables. Leucoplasts. It is colorless plastids. They have no pigment. They store starch. Example, cells of potato have lot of leucoplasts in them. Structure of plant cell central vacuole. Structure of vacuole. Vacuole are liquid, solid, filled and membrane-bound organelles. In plant cells, vacuoles are large and permanent. In animal cells, vacuoles are small in size and temporary. In mature plant cells, it occupies 90% space of cell volume. Due to its size, other organelles, including nucleus, shift towards plasma membrane. Many substances, which is important in the life of plant cell, are stored in vacuole. Functions of vacuole. They have to maintain the osmotic pressure in a cell. Vacuoles are storage sacs for liquid or solid contents. They provide turgidity and rigidity to the plant cell. In unicellular organisms, specialized vacuoles also play important role in expelling excess water and some waste from the cell. This helps the cell to perform functions like respiration, obtaining nutrition, and clearing of waste material.